let's talk about the growth of microbial populations. Most microbes are going to replicate or reproduce by binary fission. When we're talking about binary fission, this is a pretty simple, fast, rapid process of growth. And what's going to happen is that initially the genetic information has to be copied. So we will get duplication of the DNA, and then the DNA will be separated. We'll get the two copies pulled apart, and as the two copies are pulled apart, a cell membrane and a cell wall is going to be built down the middle, which will ultimately split that cell into two. So we will get two identical copies from that one original parent cell. Now, this is a fast process, and in some cases, this can be as fast as 20 minutes. So what that means is that in 20 minutes, you went from one cell to now having two cells. And then in another 20 minutes, each of those cells can divide into two new cells. So it can be something that's going to produce a lot of microbes very rapidly. The generation time is the length of time that's required for a bacterial cell to grow and then also divide, which we mean binary fission. Another way to look at this is that this is the time required for a population of cells to double in number. So if we start with one cell, one generation would produce two cells. In another generation, each of those two cells would produce two cells, and then this would continue on and on, so that at each generation we would have a doubling of what we had present. So this is known as logarithmic or exponential growth. That is what we get with microbial populations. We have this exponential growth, which can result in a lot of cells very, very rapidly. If we look at a growth curve that shows us this exponential growth, here we can see time on the bottom, and we can see that very quickly we can end up with tons of cells. We see a very rapid increase in this growth curve. It just goes up really high. So with logarithmic growth, we can basically get 2 raised to the n power of cells, and n is going to represent the number of generations that we're dealing with. To give you an idea of what we could end up with, if we assume that we have a generation time of 20 minutes, which is typical for some bacterial cells, and then we say 4 hours, and we want to know, okay, we start with one cell and we're going to give it 4 hours, and we want to see how many cells do we have at the end of 4 hours. Well, in 4 hours, we would actually have 12 generations. And if we take 2 raised to the 12th power from one cell in just 4 hours, we've got over 4,000 cells. We actually have close to 5,000 cells that are being produced from that one initial cell that we started with. So when we talk about storing food at the proper temperature, here you can see why that's the case. Because bacteria, microbes, have this capability for exponential growth. And even an hour on the counter or 2 hours on the counter, when we're done with it, can result in a lot of microbes actually growing in that potato salad or whatever it is that we're leaving improperly on the counter. A growth curve is a curve that plots the number of organisms that are growing in a population over time. And here we have a couple different ways to represent that growth curve. If you look at the first graphic, you can see that if we're just plotting the number of cells on that y-axis, we get this very sharp increase in our growth curve. And it's really difficult to see a whole lot from that just because it is such a sharp increase up. A better way to do this is to create a logarithmic scale on the y-axis. If we have a logarithmic scale, we get a nice line that shows the increasing growth of the population over time. So that tends to be the way that we draw our growth curves a lot more often, is really using the logarithmic scale on the y-axis. If we look at a bacterial growth curve, typically we can distinguish 4 distinct phases, and we can see all 4 of those phases in this graph at the bottom of the slide. In the very first phase, we do not have real growth happening. This phase is called the lag phase. So in the lag phase, typically the cells are getting adjusted to their new environment, to their new conditions. And in order to do that, they have to produce new enzymes and things that they will use to metabolize the nutrients in solution. So we will see a nice steady line when we're looking at cells that are in the lag phase. However, at some point, they will be adjusted to that environment, and at that point, we will see very rapid growth and reproduction. This is where the logarithmic growth is going to be taking place, so we will see a sharp and steady increase at that point in the number of cells that we do have in that particular population. This log phase is going to be the best phase for treatment. This is the phase that is the most susceptible to antimicrobial drugs. In the next phase, the stationary phase, this is when the cells start to die off. And they start to die off because at some point the nutrients are going to start being depleted, and the waste are also going to be accumulating. And the accumulation of waste is also going to be detrimental to the growth of the bacteria. So when we're looking at the stationary phase, we have new cells being produced at pretty much the same rate that cells are dying off. So we will once again be at a steady line, a solid line where we don't see an increase or really a decrease in our population size. And then the fourth phase is going to be the death phase. In the death phase, cells are really starting to die off, and they are dying off at a much faster rate than they were produced. So we start to see a decline in our population. This does not mean that no cells are being produced in the death phase, but we definitely have more of them dying off than we have being produced. A special type of device that we can use for populations is a Chemostat device. With this special device, we have a way of manipulating the growth of the microbes. And the reason that we can manipulate the growth is that there is kind of an intake valve where we can put fresh media in, and that would replenish any nutrients that have been depleted. And we also have a way to remove any depleted old media as well. So by carefully manipulating the media throughout our experiment, we can control exactly what stage or what phase of growth our microbes are actually in. So how can we measure microbial reproduction? Or how can we measure exactly how many cells we have present in a population? Well, there's going to be a lot of cells there in many cases, and so one way to do this is to count the number of cells in a smaller representative sample. So if we count the number of cells in a small representative sample, we can then multiply to estimate how many microbes are in the whole specimen or are in the whole sample. So one way to do that is with microscopic counts. This is basically counting using a microscope. Cells from a liquid culture are placed onto a microscope slide that has a special grid on it. And then what we do is, using a microscope, we count how many cells are in that particular square. And once we know how many cells are in that particular square, we multiply that by the number of squares that we actually have. That gives us a good estimate of how many cells we have in our initial sample. Another way is that we can use an electric counter. These are going to count cells as they interrupt an electric current. So basically you have a current or a beam that is pointing across, and then you have a device that's just going to drip those cells down. And every time a cell passes in that liquid media, the laser would detect that a cell just went by. So they're basically counting for us to help us get an estimate of how many cells we have. There are also some methods that require incubation. So the microscopic counts, the electric counts, we don't have to grow and incubate those cells in any way. If we do require incubation, typically we're getting into what we call serial dilutions and viable plate counts. So for a serial dilution, basically we take a sample, we place it into an initial amount of media, then we take a small amount of that, we put it into another fresh batch of media, and then into another, and so on. And every time we dilute it a little bit more, oftentimes this is a 10-fold dilution each time, so we may start with a 1 in 100 times dilution, then we go 1 in 100, 1 in 1,000, 1 in 10,000. So we're going to get a wide range of dilutions, and then we take that sample and we plate it out. So we put it on a petri dish, we incubate it, and we let it grow. And what we find is that we'll have a range of bacterial numbers on those plates. And we try to find one of those samples that has a number that we can actually reasonably count. So as you can see here, one plate has way too many. We can't count it. The next one has too many to count. But then by the time we get to the 1 in 1,000 fold dilution, in this case, we have a reasonable number of cells that we can actually count. So this is a good way to estimate what we have in an initial population of cells. We produce a number of different dilutions, plate it out, and then look for one that we're actually going to be able to count the numbers of. We can also do some membrane filtration. This would be a technique that's used when there's some really low numbers of microbes. For these, we have a sample that we then pour through a membrane filter, and then after we pour it through the membrane filter, we can actually place that onto a culture media like a Petri plate. And so in this way, we take a whole bunch of media and we get it reduced down to a much smaller amount. And then hopefully we'll have a few colonies that we can identify on our Petri dish after some incubation time. If we talk about indirect methods to measure population size, this would be not actually counting bacterial cells. One way that we can get an indirect measure is to look at turbidity. Turbidity is going to measure how much light passes through a culture. One way that we do this is we use a spectrophotometer. But essentially what this spectrophotometer or special instrument is doing is it's measuring cloudiness. Notice that we've got two different tubes here. One is very clear, one is cloudy. The clear one is going to have very little or no growth in it, and then the cloudy one has a lot of growth. So as we get more cells produced, they're going to be more cloudy and less light is going to get through that sample. So this is what that spectrophotometer is really doing. It is shining a beam of light through the sample, and then it has a detector on the other side. And if a lot of light gets through, then that means there's probably not much in there as far as bacterial cells go. If you look on the bottom, we have a lot of cells inside of this culture, and so when the beam of light shines through, the detector doesn't see much of that light on the other side. So that's going to tell us that we do have a greater abundance of microbes in that particular sample. So that's how the turbidity test would work. Another way that we can measure microbe population size indirectly is going to be to look at metabolic activity. With metabolic activity, we would look at how quickly nutrients are depleted or how quickly they're utilized. A third way is going to be to look at the dry weight. If we're looking at the dry weight, we would have to actually take the sample. We would have to dehydrate those bacteria. So we have to filter them out, get rid of all of the liquid, and then actually weigh on a scale how much we have there. With those, one of the downsides is that that has to be the end of the experiment. We can't continue beyond that point because we are actually removing the microbes from solution and we are dehydrating them so that we can actually quantify them in some way. We can also use genetic methods to measure our population size. When we would do that, we would actually be looking at the abundance of specific unique DNA sequences. And PCR, or the polymerase chain reaction, is one way to look at the abundance of certain unique sequences.